Okay, so the very first phylum that we're going to look at is the Phylum Porifera. And this literally means pore bearing. These organisms are filter feeders. And so it is through these pores that water travels through the sponge. So if I look at this, this looks like a mouth, but sponges do not have mouths. They actually don't even have tissues. They actually don't even have digestive system. So their level of organization is that they are only aggregations of cells. So they are not eumetazoans. They are just simply aggregations of cells. So they have no tissues. Okay, no tissues. So they don't have a nervous tissue. They don't have muscular tissue. They are essentially just sessile, except when we'll look at the larvae. They're sessile, and they just sit there and pump water. So what this is up here is called the osculum. And the osculum is an opening. Water actually travels through pores in the body, and then it travels up and out through the osculum. So there's this current of water traveling across cells that then specialize in capturing food and other things from the water. So let's look at these specialized cells within our sponge. So notice that we have cells that make up the pores, and water is coming in through those cells. Those cells are called porocytes. Lining the inside, and this is a very simple sponge, but some sponges are like branched. Lining the inside, we have a special cell called a choanocyte. So choanocytes are actually collar cells. So these are called collar cells. And I apologize for my pen is just not responding the way I want it to. Collar cells. And these have this collar. Water moves across it, and it is very much like a sieve or a net. So food particles get caught in the collar cells. The collar cells are capable of intracellular digestion. So they take food in, and this type of digestion is called phagocytosis. So phage means food, and it's taking it in. And it is then going to pass it to another type of cell called an amoebocyte. And the amoebocyte is then going to carry that food throughout the rest of the sponge. And it is going to transport that food and feed all of the other cells, like the pore cells that need food as well. So we do have other types of cells. There's another type of cell that produces the spicules, and the spicules are actually the skeletal elements of the sponge. So some sponges have soft spicules. The bath sponge that you might get at the grocery store, a loofah, that is actually not a sponge, that is actually a plant. The sponges that you get at the art store that are weird shaped, those are actually sponges. And those sponges have spongin as their spicules, which is a protein. Sometimes the spicules can be made of calcium carbonate so that they're hard. Sometimes they can be made of silica. We have glass sponges. But the spicules are the skeletal elements, and they are not cellular. So they're just made of different types of organic molecules. So these are my skeletal elements of my sponge. Okay, so we have these different cells making up this organism. One characteristic of an organism is that it must be capable of reproducing. And so sponges can actually reproduce asexually as well as sexually. So let's look at asexual reproduction. So this means that you are producing an organism that is identical to the single adult. So it is like cloning. Sponges, when they are dehydrated, when the water starts to dry out, and some sponges are freshwater, but there's a lot of marine sponges, the big ones are marine. But when it starts to dry out, they form packets of amoebocytes called gemmules. So gemmules, because I'll show you in a minute, they look like gems. So gemmules are packets of amoebocytes. Amoebocytes. So this is when the environment starts to dry out, and this allows them to survive drying out, because when the water comes back, then these gemmules, these burst open, and one amoebocyte can give rise to a whole new sponge. So that is some pretty amazing regenerative ability. And this idea that you could have like a stem cell, like a cell that is undifferentiated, that could give rise to a whole new individual. It'd be like if you took a liver cell from me and then you grew a whole new me. This has a name, and it is called totipotency. So that means total potential is in this one amoebocyte, totipotency. So the amoebocyte exhibits totipotency. Okay. When we look at sexual reproduction, we have the formation of egg and sperm. And these egg and sperm then recombine to produce organisms, that offspring, that are genetically different from each parent. And so we have the production of egg and sperm. Now the egg and the sperm are produced inside of the sponge. The sperm are released, but the eggs stay inside of the sponge. So sperm are released into the water. So the question is, how do they get to another sponge to fertilize the egg? So my sponge is filtering water, not just of food, but of sperm. So you can imagine that the oceans are filled with eggs and sperm, because a lot of organisms just release that in mass, right? And so if it is the sperm of a species that is not of the right kind, they could just use it as food. But let's say that this sponge captures in its choanocyte the sperm of the right species. And it might be able to tell that because of chemical signals on the surface of the sperm cell. It then has to transport that sperm to the amoebocyte, and the amoebocyte delivers it to the egg inside of the sponge. And that is where fertilization takes place. This is weird. Like, I would think that it would, if a sponge is so simple, they would just release both egg and sperm into the ocean, like, say, for example, jellyfish do. But that is not the way it works. So it's a little bit complex for such a simple organism. So the offspring that are produced are mobile larvae that can swim around. So they can then go to a new environment, settle down, and become an entirely new sponge. And we oftentimes see this in animal life cycles in terms of asexual and sexual reproduction. Sexual reproduction usually produces larvae that can disperse. Because they're genetically different, perhaps they would go out into the environment and find the best spot to set down. If we're talking about asexual reproduction, the adult is doing pretty well in its environment if it can reproduce. So its offspring tend to stick around in that same spot. So let's look at some images of this. This is my multicellular embryo that is released by the sponge after fertilization takes place. And you can see that it has cilia and that it is swimming around. This looks very similar to, early embryonic development in almost all animals. This is my gemule, and this is what they look like. The gemmules, these are actually the spicules. And so the gemmules are formed when the sponge dies, but it leaves these packets of amoebocytes that can withstand dehydration. And so you put these in water and they can become entirely new sponges. Sponges also are kind of known for their ability to regenerate. So you can do experiments with them where you take a sponge and you put it in a blender and you just and separate out all the cells, and then they can regenerate into new sponges. And so artificially, we can create new sponges by separating out the cells.